This is the Naked Genetics podcast, taking a look inside your genes. It's the hottest new biotechnology technique to hit the headlines since ever. Crispr is a precision set of genome editing tools enabling scientists to cut and paste together DNA in any organism exactly how they want. And the implications for human health and even humanity are huge.
What you end up with is, in effect, this sequence that you've added precisely inserted in the targeted position in the genome.
Plus, linking genetics to lifestyle, and our gene of the month is black and white and very cute. This is the Naked Genetics podcast for February 2016 with me, Dr. Kat Arney, brought to you in association with the Genetics Society, online at genetics.org.uk. The speed at which CRISPR has moved from the pages of dry academic journals to the headlines of newspapers, not to mention labs and biotech companies around the world, has been breathtaking. This month, the UK Human Fertilisation and Embryology Authority granted a licence for scientists at the Francis Crick Institute in London to start using the technique to modify early-stage human embryos, although these wouldn't be allowed to grow past the stage of being a tiny ball of cells. Although it might seem like the CRISPR story has come out of nowhere in the past year, the Naked Genetics podcast was on the case back in 2012 when University of California, Berkeley researcher Jennifer Doudna and her collaborator Emmanuel Charpentier, who was at Umeå University in Sweden, published a paper in the journal Science entitled "A Programmable Dual RNA-Guided DNA Endonuclease in Adaptive Bacterial Immunity." Here's a clip from the podcast back in July 2012, where science writer Nell Barry and I discussed the implications of their discovery. I love this just because of the potential of this story. And this is researchers who've been looking at bacteria and looking at how bacteria use certain molecules like molecular scissors to snip up their DNA and glue it together, and helps them develop different characteristics to help them survive. But they've actually looked at these molecular scissors and found out how they work. And they think that possibly you could use this technology to make programmable scissors. So you could cut up human DNA, animal DNA, and basically glue it back together in any kind of way you like.
That's pretty cool. I mean, I think it's kind of what I think of as genetic engineering. I guess you think, you'll just put together whichever bit of the DNA that you want. But actually, in reality, that's not really possible yet. So I suppose this is taking us a little bit closer to real designer genetic tailoring, or whatever you might like to call it.
So I remember when I was in the lab, and this was some time ago now, so things may have changed. But if you wanted to do genetic engineering and stick different bits of DNA together, you had to look at the sequence, and there were only certain sequences that you could cut using enzymes. Things have probably moved on now, but I think there is some restriction in the sequences that you can cut and glue together. So having a different set of technology could really be exciting.
Yeah, and I guess the sort of eventual implications of that would be that you could create these kind of designer organisms, like maybe bacteria that degrade nasty environmental toxins or produce things that we need for drugs, or all kinds of exciting applications, because we know that bacteria and fungi have all these abilities, and it's just about, could we harness them to do the things we want, I guess.
Watch this space, I think, because it is still very early days for that. But thank you very much. That's Nell Barry, science writer. Let's take a closer look at exactly what's involved in CRISPR, or to give it its full name, CRISPR-Cas9. To find out more about this molecular toolkit and what it can do, I went along to a meeting organised by the Progress Educational Trust, discussing new genetic techniques and the impact they could have on human health and human genomes in the future. I caught up with one of the speakers, Tony Perry from the University of Bath, who told me more about the story of CRISPR and some of its potential applications.
The prototype comes from bacteria that give you a sore throat, actually. The sore throat bacteria, Streptococcus pyogenes. And it seems to be a kind of defence system. I mean, it turns out that even bugs get bugs. And so these Streptococcus pyogenes, these sore throat bacteria, themselves get invaded by viruses. And the CRISPR-Cas9 was evolved as a way of cutting the viral DNA and thereby defending themselves, the strep pyogenes, against these viral invaders.
So what are the components of this self-defence system? How does it work, and how have researchers then hacked this to do more precise editing?
The system really is like a pair of molecular scissors and a molecular sat-nav. The sat-nav is a small RNA molecule, and it guides the scissors, the molecular scissors, to precisely where you want it to go in the genome. And the reason it does that is because, like a sat-nav, it's programmable. So you get to tell the sat-nav what the coordinates are in the genome, and it will then take the scissors to that point in the genome, and the scissors will cut there. And when they do so, when the scissors do so, they make a double-stranded break called a DSB. And then the cell has got two types of toolkit that it can use to repair that double-stranded break. And one type of repair that the cell can use, or one toolkit, is a simple kind of paste mechanism.
Just get the ends and glue them back together again.
Absolutely, just get the ends, glue them back together again quickly, because it does have to be a rapid process, because these breaks are anathema to cells. If they're not repaired quickly, the cell can die, and even worse, it can give rise to cancer and death of the organism in multicellular organisms. So although it's very useful for research, because you can use it to knock out genes if the cut is in a gene, it's probably not going to be so useful for clinical applications. Anyway, and the second toolkit that the cell has, or mammalian cell has, to repair a double-stranded break is called homology-directed repair. Well, we can actually add in our own designer piece of DNA, which is used for repair, and so we can design that. We can produce it, fabricate it in vitro in the laboratory, and make sure by sequencing that it's exactly what we want. And then we can introduce it into the cell at the same time as we've introduced the molecular sat-nav and the molecular scissors, the Crispr-Cas9. And so the cell, in the right circumstances, will use this extra piece of DNA that we've added as a repair template. And what you end up with is, in effect, this sequence that you've added precisely inserted in the targeted position in the genome, so that it's been used to, in other words, to repair the genome, and you've introduced the change that corresponds to the DNA that you've added in.
So we've got the CRISPR, which is the RNA, the guiding system. We've got the Cas9, the scissors, and then this DNA template, and that could be anything I can imagine.
There can't quite be anything you can imagine as yet. Perhaps one day. The constraint, one constraint at the moment, is the size that you want to. If you want to introduce a piece of DNA, so you make the cut where you want the cut to be. So you can introduce a large piece of DNA where you want it to be. But there probably is an upper limit at the moment today on how much novel DNA you could introduce, and we're probably in the many thousands of base pairs, or many thousands of characters in the genome, and we don't know how, whether the sky's the limit or not. But what we do know is that as you increase the length of information that you want to introduce, the efficiency drops. And so probably if you wanted to introduce a really large piece of DNA, for whatever reason, you might have to do that in multiple steps.
So we've got this technology which seems like an incredibly powerful tool, and I know that scientists all over the world are using it in all kinds of ways in research in the lab to understand how genes work and how genes get turned on and off and all this kind of thing. But what we're talking about at this meeting here is editing the human genome. Tell me about the two different approaches that people are trying to think about, not doing necessarily at the moment, but the two sort of different ways that people are thinking about doing this at the moment.
So as I see it, this whole topic can be sort of perhaps rather artificially, but sorted into whether the changes that you make to the genome are heritable, or in other words, they can be passed on in successive generations, or are not heritable. So the two main types of non-heritable genome editing changes that you might make and work together with existing technologies that are variously well established.
So this is effectively just normal cells of the body that are broken. You're repairing them in some way.
You can repair cells in situ using gene therapy, and this would be coupled together very nicely with Crispr-Cas9, but you here are doing the editing, as it were, of cells in the patient, of the patient cells in situ.